Just how it is. I mean, it's not gonna change. It's just that I don't want it to. I just don't want it to seem like I'm just being a weird ass.
Okay. Is your name on it? And all that? All of you. I know most of you. So, yeah, go.
It took me 10 minutes to the library and their printers are doing it. Is it okay if I submit it just as a document and then print it out?
Did I say that you're supposed to do that?
Yeah.
Okay, no, I couldn't remember what I said. That's fine.
Okay.
I appreciate your efforts. Okay, that is right. That is right. We've got one more minute. Oh my gosh. I forgot my binder. Did I really? I really forgot my binder. Okay, there you go. There's something for you to think about while I do my binder. you all done? No. One more minute. Either way. I know most people, so I'm still. Okay. Before we talk about this, let's talk about the outlines. So I think I might have told you you need to turn in online and a hard copy. I don't know.
I mean, there was a turn it in online, so I.
Okay, so what I'm going to say is, if you turn in a copy either way today, I'm okay with it. Okay?
What do you normally want?
I'm okay with you just turning it in online. That's okay with me. I think I posted links in the next couple weeks, thinking that's what you would do. But then people were giving me hard copies, so I was like, did I say something else in the syllabus? Which I did read the syllabus at some point in December, but maybe not since then. So I'm sorry for the confusion. If you're a person who likes to turn in hard copies, I'm okay with that. I'm a person who likes to grade hard copies. But if you would like to just turn it in on Moodle because there's a link there and that's the way you like to do it, I'm okay with that. Again, I'm a person that believes that writing things helps more than typing things sometimes. So if you like to write your outlines, that's fine with me. If you want to turn in a hard copy, I'm going to grade them pretty quickly and get them back to you. At least that's the idea. Okay, I talked to one person over email, so I didn't really talk. You know how much I don't know what kind of feedback you got last semester on outlines. I'm not sure. So my thinking is of how I'm going to do it. It's going to be out of 5. So 5 would be like, wow, you did everything really great. 4 would be like, wow, you did most things really great. 3 would be like, well, you did the stuff adequately, maybe-ish. 2 would be, well, okay. And 1 would be, well, you turned something in. And 0 would be, you didn't turn something in. Okay, so if you have filled in everything completely, it's going to be a 5. If you filled in most stuff, it's going to be a 4. If you filled in some stuff, a 3. If you sort of attempted a little bit, it's going to be a 2. And if you gave me something, it's going to be a 1. Okay? So, also, I don't think I mentioned this in lab last week because we weren't in our normal lab. I think everybody was in lecture in lab, but you will have a folder that in the lab. So in our normal lab in 214, there's a crate of folders. So when I grade these, I will put them in your folder if you gave me a hard copy. If you didn't give me a hard copy, there will be a grade on Moodle. I don't use the Moodle gradebook, and I don't think Dr. Sayer used the Moodle gradebook. So don't ever look at the Moodle gradebook and think, gosh, this is my grade, because it's not. Okay? The reason I hate the Moodle gradebook is it won't let me do half points, and I like half points, not quarter points, not 3/4 points, I like half points. So if I want to give you like a 4.5 on an outline, I can't do that on Moodle, and it makes me crazy. So that, like the Moodle gradebook, there is the Excel spreadsheet which you can use on Moodle and track your own grade, and you're definitely welcome to do that. But the Moodle will show you a grade that is not your grade.
This week we're still in 216, right, for lab?
This week we're still in 210, yes. I said 216, it was 216. I only know like 3 room numbers, and it's 210, 214, and 114. Okay? Other questions about scheduling kind of stuff? I did look over the rest of the syllabus for up until the next exam, and I think the exam's going to be on Friday the 4th, based on what I looked at. Okay, I posted that on Moodle. I also posted a little bit on Moodle for that week of this is what it's going to be on, except I didn't put any details for chapter 27 and 28 yet, because I kind of want to feel out how we get through that. Okay? Okay. Any other scheduling questions? Let's talk about this quickly, and then we're going to dive into chapter 19/26. 26/19. Okay, so reviewing from Friday, we talked about the idea of positive control for the lac operon. When glucose is low and lactose is present, the lac operon will be turned. If glucose is low and lactose is present, then the lac operon will be turned on. Which kind of operon is the lac operon? Inducible. So it's induced by the presence of lactose if glucose is low. So that I just kind of did the second part there. When glucose is high and lactose is present, the lac operon will be turned off. And actually, that's not even really accurate. I debated whether the word turned or not. It's not really turned off. It's just off. It's already off. But even though that lactose is present, if glucose is high, it's not going to be turned on. The next one says, give an example of how the gene expression can be regulated at each level in eukaryotic cells. So at the DNA level, what's one way that we can regulate gene expression? Sorry. Go ahead. So what do you mean by that? I don't disagree.
So, like, depending on how tightly or loosely the histones are packed together, like the ones that are more tightly packed together is the DNA that's less, it is used less often?
It's less accessible because the machinery can't get in there. So the nuclear architecture, the packing of the chromatin or histones and modifications to the histones can all control how well the RNA polymerase and all the transcription factors can get in there to start transcription. How about at the transcription level?
Talking about transcription.
At the transcription level, how can eukaryotic cells regulate the process? Remember, when we say regulate expression, it can be enhance or repress, either way.
Enhancers with like control elements.
Thank you. Yes, the control elements and the enhancers are those proteins that help to bind to the control elements and make the transcription initiation complex form better. So that's the way gene expression can be enhanced in eukaryotic cells at the transcription level. How about post-transcription? We didn't talk about this in detail. We just kind of said, you can do a couple things real fast. I said it. So the way the RNA is processed can help affect gene transcription, the 5' cap, the poly A tail, the way that the mRNA is spliced, and also the stability of the mRNA. Is it going to last a long time? Is it not going to last a long time? The longer it lasts, the more it's going to be transcribed. Or translated, sorry, because it's already been transcribed. How about at the translation level?
I think we stopped at transcription.
Oh, we went all the way. We didn't talk about a huge lot of detail here, but Jonathan, I think you said something.
Cleavage.
What?
Cleavage.
Cleavage. So the way that the protein is cut, I would put that under post-translation, but yes. So remember, proinsulin is formed as a protein. It's translated as a protein as proinsulin, but it has to be cleaved in order to make the insulin active. I don't know if we talked about a lot of specific examples of translation. I think we talked about the fact that you can have global translation, like the translation can be turned off completely. Like plant cells that are put in the dark will not transcribe or not translate certain mRNAs. So mRNA can actually be stored until it needs to be translated. So plants that are put in the dark don't translate the proteins to make things like chlorophyll because they're not able to go through photosynthesis without light. So you can globally turn off translation or specific translation. I think we just said you can turn off specific translation of specific proteins. Any questions?
We talked about assembly and modification. Would that go in?
I'd say assembly and modification are probably post-translational ways to control gene expression. So you can modify proteins after they have been made to make them functional, where they go in the cell, and also affect how they're expressed. Okay, so now I want to say we're doing a whole switch 180, but we're not quite doing a 180. We're going to kind of launch into our survey of life. Okay, so this was kind of a leftover from Bio 111. For Bio 112, it's a survey of life, except we don't really talk about plants. So it's a survey of life except plants. So those of you who are straight biology majors, you're not biomedical sciences and you're not biochemistry, straight biology majors, you'll take another semester of biology. I know, how fun. It's called Bio 113, which covers plants, ecology, and evolution. Those of you who are not biology majors, somehow, if you're biochemistry or if you're a biomedical scientist, you missed plants. I'm sorry. It's disappointing, I know. You did talk about photosynthesis, but you're not going to get all the different kinds of plant survey kind of stuff. So we're actually going to skip to chapter 26. Oh, whoops, there's the answer for that. I didn't put all the answers for the other one. So we're skipping to 26, phylogeny and the tree of life. This is all we're going to talk about with 26. I think there's a little bit on an outline of 26.
Yeah.
There's not an outline for 19.
It's on this one.
It's on 18? Or it's on 26?
No, so there's 26, 19,
Okay. So the next one's a big one. The next outline has 19 on it. You can see it looks like that carefully too. All right. Okay, so there's a little bit on 26. You don't need to read the whole chapter. What we're talking about in class is what you need to know about this. So we're talking about phylogeny and the tree of life, and this is a cute little picture of snake. Wrong. It's not a snake.
It's a salamander.
It's not a salamander either. It's not a worm either. It's a lizard.
Without legs.
It's cute though, right? It's cute. Okay, and that was kind of a trick question. I wanted you to say snake, so thank you to everyone who said snake. It looks like a snake. Okay, when we talk about phylogeny and the tree of life, we're talking about the way that we're classifying animals. Okay, and so we're classifying animals based on characteristics. And when you look at this, and I will say I'm with you, when you look at that, your first instinct is that's a snake. Why do you think it's a snake?
It looks like a snake.
Why does it look like a snake?
They got no legs.
It doesn't have legs. What else?
Scales.
It's scaly. Okay, it looks like a reptile and it doesn't have legs, so you think, gosh, it's probably a snake. And I get that. And again, it was a trick question. So let's talk about why it is not a snake. This is actually a European glass lizard. And there are reasons that it's not classified as a snake. And again, when we talk about phylogeny and classification, as biologists, we're looking at what are the characteristics of an organism and how are we going to classify it. And sometimes we change our minds. Like you think you know something and then you don't actually know something. So this is not a snake. So we have a picture here. Why is this not doing this? This is a snake. On this side, no, I haven't fixed the stylish issue yet. And this is a glass lizard on this side. So there are some things that are different. The jaw structure of a snake and a lizard are different. Snakes have what we call, and I really like this term, a kinetic jaw. Okay, as you may know, a lot of snakes swallow things that are way bigger than their mouth. That's because they can actually unhook their mandible to open their mouth bigger to swallow things that are very large. So a snake has a kinetic jaw. A lizard does not have a kinetic jaw. Tails. Snakes have a tail. This is also, I don't know what they have a post-anal tail. So past their anus, there's a tail. This lizard does not have that. I'm not going to be able to fit it all. They don't have a tail past there. It doesn't have a tail past its anus.
Do you want us to write this down?
It's not going to hurt you to write it down. Okay, their scale structure is actually different. The way that the scales specifically on their belly, we're not going to go into all the details of it, but the scales on their belly are different because snakes and the lizards burrow. They're burrowing, like they make holes. I think snakes just use other people's holes. I don't know if they make their own holes. Their tongue structure is actually different also. Lizards have holes on the outside of their body for ears, and snakes don't. Snakes don't have eyelids, and lizards do have eyelids. So all of these would be characteristics if you found something that you thought was a snake, and you knew it wasn't a poisonous snake. you could look at to say, is it a snake or is it a lizard? Okay, life on Earth is very diverse. There's all kinds of crazy things out there. There are things that are very clearly in categories, and then there are things that are kind of, maybe they fit in a category, and maybe they don't fit in a category, and things aren't always what they seem. So when we talk about classification, it's always changing. So we say we classify something as a this or a that, but as we get new information, we might change our minds. One of my favorite teaching moments was the year I told my students not to use Wikipedia because it's wrong sometimes. So I pulled up the slide, and we were talking about classification, and I was like, look at this slide, here's a panda bear, and it says it's in the bear family, and panda bears are not bears, they're more closely related to raccoons, and this is why you can't trust Wikipedia. So I was telling one of my friends about it, he's like, Rachel, actually they did reclassify panda bears as bears. And I was like, wait, what? He like sent me this scientific journal article that talked about we have new information, and based on the new information, the DNA information that we've gotten, they've reclassified panda bears as bears. So I had to go back to my class and be like, so remember how Wikipedia is sometimes wrong? Sometimes your teacher's wrong too. And guess what? Panda bears are bears. So yeah, we change our classification based on new information. And DNA sequencing has really changed the way that we look at relationships between organisms and put them into different groups. So, when we talk about phylogeny, the specific definition of phylogeny, I don't like the color red, is this is the hypothesized evolutionary history of a species. I'm not going to write that all up here because you know I can't write very well still. That says evolutionary history. And when I say hypothesized, what does that mean? This is what we think, based on what we know right now. It's not like we're just randomly guessing, but we have information and this is what we have, this is what we think, but we might change our minds based on new information. It's a hypothesis still. Okay, so we use things like fossil records, the anatomy, like things like a kinetic jaw versus not a kinetic jaw, genetic information, molecular information, all that goes into understanding the phylogeny of an organism. So this also shows us relationships between organisms. And it can be depicted in a lot of different ways. So this is showing you two different ways. Maybe now, okay, there we go. So over here on this side, this is the Linnaean, like classical Linnaean classification. Carl Linnaeus was the guy who kind of came up with this idea of let's organize things based on different levels. And each one of these different levels are taxons. That is not very well written. So you can see here, this is showing you the full classification, Linnaean classification of a panther. I think that might be a cheetah. Of a large cat, whose scientific name is Panthera pardus, which I should have looked up. Okay, so you can see here, starting at the bottom, we're starting at the largest level of classification, and you probably have talked about this before. There are 3 domains of life. There are bacteria, which we're going to spend a lot of time on the next few days. There's the archaea, which they're just kind of like weird bacteria organisms, weird prokaryotic organisms, I shouldn't say bacteria. And then eukarya, which are all the eukaryotic cells. Okay, so there's 3 domains, so everything that's alive fits into one of these 3 domains. From there, the domains are divided into kingdoms. I don't know how many kingdoms you learned. For eukarya, there's 4 that I know of. I used to ask this question when I had parent night, and some people would say 2, and I was like, now I know how old you are. You think there's only 2 kingdoms, haha. Okay, so animals then, we go to phylums, and we'll spend a lot of time going through the phylums. Chordata are the ones that are mostly vertebrate-ish. So those are the things that people like to talk about, but we're going to talk about all of them. All the invertebrate phylum. If you've ever watched Finding Nemo, which if you haven't, you should. Mr. Ray sings a lovely song that talk about a lot of the phylums. I'm not going to sing it for you now. I'll find it for you when we get to it more. But he talks about like Peripherenidaria, Hydrozoa, which is actually a class, so that bothers me because it's not the right level. But so phylums, then class, then order, then family, then genus, then species names. You probably learned some kind of saying for this, maybe, like King Philip came over for grape soda or something like that. Nothing. Yeah, something like that might ring your, might remember that from high school biology, middle school biology even. Okay, so each level just gets more and more specific, and we're learning more and more specific characteristics. This species name, I'm going to talk about it now, and we're going to keep talking about it because I'm going to be picky about it. This, when we write a species name, that's not very clear because I'm on the edge. We use binomial nomenclature. Binomial means how many names? 2. You always write the genus name and the species name when you're talking about the scientific name of an organism. What do you notice about the genus name? I crossed it out, number 1, accidentally. Panthera, how do we write it? With a capital P. And the species name we write with a lowercase letter, a little p. And the whole thing is in italics. But guess what? You can't write in italics. You can type in italics, but you can't write in italics, so what do you need to do? Underline it. Capital genus, lowercase species, underline the whole thing. And again, Carl Linnaeus came up with these ideas. The idea of this is that we have standardized communication. So if I'm writing a paper about the Panthera pardus in English, but my friend in India wants to learn about Panthera pardus also and wants to read my paper, I've never written a paper about Panthera pardus, we would be talking about the same thing because we're using the scientific name. Whereas common names, there's lots of different things that have more than one common name. Am I able to come up with one off the top of my head? No. Why not? I don't know. Okay, so those little things that look like lobsters that hide under rocks. I heard crawdad. I heard crayfish.
Wait, no, okay, they hide under
They look like lobsters. They're not salamanders. I like crayfish. Crayfish. Some people call them crawdads, crawdaddies. I've heard all kinds of different names for them. But if we were going to talk about it in a scientific way, we use the scientific name, we'd all be talking about the little lobster things that hide under rocks in the creek. Okay? And that's why we have a standardized communication way, a standardized scientific naming process so that we can all talk about the same thing and mean the same thing. Darius.
I'm sorry, it's binomial what?
Nomenclature. You don't have to be sorry. I should be sorry. Okay, sometimes we write it more like a tree over here. This is called a phylogenetic tree. And again, if you take 113, you'll spend lots of time talking about phylogenetic trees. Again, this is showing the relationships. Here's an ancestral lizard, and then it splits off into this group, which keeps splitting, and the geckos. So each one of these are like a branch in a family tree. So each branch is a common ancestor, and each group, like these guys, is what we call a clade. So it's a group of related organisms, and they're with a single common ancestor. So both monitor lizards and glass lizards came off this common ancestor here, which we don't get the name of. But
You said clade is sharing common ancestors?
yes, so it's a group of organisms that share, they're a related branch of organisms that share a common ancestor. So we draw lots of these pretty phylogenetic trees with nice organized patterns, and the lines all are parallel, and it looks really pretty. In reality, up there it's circled. Oh. A phylogenetic tree probably looks a little bit more like this. It's messy. Like a lot of family trees. It's messy. Okay, again, these three, whoops, nope. These three are the three domains. So we're going to be talking about all three domains. You need to know there are three domains: eukarya, archaea, and the bacteria domain. We're going to start down here with bacteria, and we're going to work our way up to the Animalia, and all through the different phyla of the Animalia. But we're going to start today in chapter 19 with, I have Dr. Smithers' notes and they make me laugh. Some of you didn't know Dr. Smithers, but they make me laugh. We're going to talk about some weirdos.
Chapter 19 or 26?
Chapter 19. This is the end of chapter 26. Now we're going to chapter 19 to talk about the weirdos that don't really fit into this phylogenetic tree. Viruses. Yes.
So you said earlier that there are 14 of them?
Within eukarya, yes. Protists are not on there. I don't know why. Protists? I don't know why. I noticed that too. Okay. So, as if we haven't heard enough about viruses in the past 2 years, we're now going to start talking about chapter 19. And why do I say that viruses are weird and they don't fit into our three domains even? Why are viruses weird? They're not what?
They're not, they're not the same as certain
Why? I agree.
mutations.
But other things can mutate too. Why are we saying, so we're talking about a survey of living things, and I'm saying we're starting with something weird, viruses. Why are viruses weird when I say we're talking about living things? Tori?
They don't meet all the criteria.
They don't meet all the criteria of life. So again, sorry, a little throwback to 111. You talked about all these characteristics of living things. Viruses don't meet those. Specifically, what do viruses not do that make them not meet those criteria? There's two major criteria that they don't meet. What can they not do that living things have to be able to do? They cannot reproduce on their own. So they can't reproduce on their own. That's not to say they can't reproduce, but what do they depend on? They need a host. So they can reproduce with the help of a host, but they can't reproduce on their own. They also can't metabolize on their own. So getting energy is not something they do on their own. They need a host. So they're not quite living, but they're not really dead. So they're kind of in this weird zone, and I think your book uses the phrase, they live a borrowed life. Are they zombies? But we can kill them. So they're not really zombies because we can you kill a zombie? I don't know. We're not going to go down that road. Okay, they are what we call obligate, intracellular parasites. They have to have a host to live. They're obligate, meaning they are obligated to have a host. Intracellular, meaning they live within the cells of other things. And they're parasites, meaning they suck things away from cells. They take away from their host. Do they kill their host? Sometimes. Sometimes not. All of you, I'm sure at some point, have survived a viral attack. So not all viruses kill their hosts, thankfully. So this is showing you some pictures of a virus. This is HIV. So this over here, all these little red dots that you can't see, I am putting blue dots over them. Those are viruses. This is a CD4 T cell. This is a specific kind of cell that is infected by the HIV virus. Okay, so all of those are new little viruses being formed. When we talk about viruses, there's two parts to a virus. All viruses have, I should have put a blank slide in. Can I put a blank slide in? No. It's not going to let me do that. Okay. So when we talk, there's two parts. What are the two parts of a virus? There's the capsid. So you see all of these have a capsid. They're not showing it. They didn't label it on these, but the capsid is a protein coat. Oh my gosh. That's a T. I can't go back and fix things well either. The capsid is a protein coat. What's inside the protein coat? DNA or RNA, so some kind of genetic material. So a virus is essentially some kind of genetic material surrounded by a protein coat. And it infects a host cell in order to be able to survive and reproduce. Oh my gosh. So when we talk about that genetic information, it's important that you understand that genetic information can be DNA. It can be RNA. It can be double-stranded of either one, and it can be single-stranded of either one. And again, I think you guys probably have more background talking about viruses than 2 years ago we would have had for sure. So it's DNA or RNA, it's double-stranded or single-stranded, or it can be linear, or it can be circular, and it can be really, really super small. Some have 3 genes. Some viruses have 3 genes, up to thousands. Most of them are more in the middle range, 50 to 100 probably, 50 to 200. But they're as small as 3 and as big as thousands. A lot of viruses we name, or viruses are categorized based on what kind of genetic material they have. So we'll say, oh, that's a DNA virus, that's an RNA virus. What's COVID? It's an RNA virus. Okay, and then we also look at the shape of the capsid to help be able to classify bacteria. So you can see here, here's a couple different shapes. This is the first virus that was discovered, the tobacco mosaic virus. So it infects. What do you think it infects? Tobacco. Wasn't a trick question. I know, I got you all in the lizard thing now. Everybody's hesitant. It infects tobacco, and it's called mosaic virus because it makes the leaf have different colors. Where it's infected, it will turn different colors. And so this was the first virus that was discovered in the late 1800s. It wasn't actually, they didn't actually figure out it was a virus until 1935. But scientists were able to take the sap from plants that were infected by tobacco mosaic virus, and they would put it on another plant. They would get it, so they thought it was a bacteria, but they couldn't see it under the microscope because they're smaller than bacteria even. And so it took probably 40 to 50 years to figure out that it was a virus because they couldn't figure out that it was something different than a bacteria because there weren't words for it yet.
Who found it?
What?
Who found it?
Who's the first person, or who's the person that said it was a virus?
This is the person who found it. Who actually tried to dig it out in question what.
It was a German person whose name I don't remember. Sorry. It's in the book.
It is?
Yeah. That's another. Last name is in your book. It was a German scientist who took the sap originally from a plant and rubbed it on another plant and said, hey, this plant got sick too, so there must be some kind of infectious agent, and it's probably bacteria. Then the next guy who did a little more work on it passed that sap through a filter that was small enough to filter out bacteria, and then rubbed it on another plant, and the plant got sick still. So he thought, oh, it must be a smaller bacteria than we've ever seen before, but it's still bacteria because, again, no more words. Virus wasn't a word yet. And then in 1935, the guy, who was Dutch, who figured out that it was a virus, actually like crystallized the sap and like crystallized the virus and said living cells don't crystallize, so this has to be something different. So we can't make living cells form crystals, but we form crystals of this tobacco mosaic virus, So that's when the idea of this being a virus came about. So you can see here, it's an RNA virus, and its capsid is linear. I don't know, linear is not the right way. It's arranged it's actually arranged in a helix. So it's what we call a helical virus. So these protein molecules go around and around and around and around and around and around the RNA. And the RNA is wound up inside there. So that's a helical virus. There are not a lot of different kinds of protein in the capsid for a bacteria. There's lots of different proteins that could be involved, but like the tobacco mosaic capsid is only made out of one kind of protein. So they tend to be pretty simple.
When you say that the virus could be crystallized, what sort of, like how would that manifest in the virus? Would the DNA change shape? Would the capsid change shape?
It just forms a regular crystalline pattern when it gets like treated with chemicals. That's about it. I didn't read a whole bunch about the experimental procedure, but they were able to crystallize it out so it forms regular crystals. So it's not that it would in like a cell or anything do that. That's just the way that they processed it, that they were able to figure out. They processed the sap and made these crystalline things, but then they could use the crystals to infect other things. I don't know. Okay, adenoviruses you might be somewhat familiar with because those cause things like the cold. You can see it's got a polyhedral shape on its capsid. It's showing you a DNA virus here. And again, these sizes are showing you how tiny we're talking about. 18 by 250 nanometers. A nanometer is 10 to the negative 9th of a meter. Influenza virus, we're starting to add something extra when we talk about things like the influenza virus. You'll see here some viruses have an envelope around them. And that's really important in the way that it causes infection and the way that it is involved in the way the host cells are involved. This is actually, the envelope is actually a lipid membrane. And guess where they get it? From the host. So the lipids are stolen from the host. Are they evil or are they genius? Are they evil geniuses? Okay, it's a lot easier to look like something if you take its membrane. And then you'll see these spikes. Again, spike proteins. Have we heard that term recently? Yeah. The spike proteins on the outside. So it's a combination of host membrane and viral information like the spike proteins. The last category of bacteria viruses on here are the bacteriophages. These are like the creepy alien-looking ones. I'm always a little disappointed when I talk about them because they do not infect humans. They only infect bacteria. So you will never be infected by a little creepy alien bacteria. Virus, sorry. They only infect bacteria.
Do they infect bacteria that are found in the body?
Yeah, so they could infect, yes, they can infect bacteria that are within your body. So technically, yes.
What if you had bacteria that infect the lactobacillus that are found within the body? Wouldn't that mess up
So
the microbial?
yes. So one of the things that I was reading about bacteriophages is you have E. coli in your body all the time and it doesn't bother you, but when you get food poisoning from E. coli, which is like E. coli 0157 or something, that's one that's been infected. And they think that might be why. So again, we're not going to go into tons and tons of detail about all of this. We're going to talk a little bit about how viruses reproduce in a generalized kind of way. And then we're going to talk about a couple of viruses, and then we're going to be done with this. Okay, so this is a generic replication of a bacteria. This comes from your book, and I forget which figure it is. It's before 19.7. Okay, again, replication has to happen in a host. So here's our host cell, and here's our virus. What kind of virus is this? It's a DNA virus. We can't say for sure anything more than that. It's a DNA virus. Again, that's how we classify it, primarily based on its genetic material. So the first thing that has to happen in order for it to be able to infect a cell is it has to get into the cell. So the virus is going to enter the cell, and the uncoating happens, meaning the capsid is going to be broken down so that the viral DNA is exposed. Okay, so this is a DNA virus. So when it's a DNA virus, the cell is just generously going to give of its enzymes that are needed for replication, and it's going to make more viral DNA. So just like it replicates its own DNA, it's going to replicate viral DNA. Okay, viruses don't have those enzymes that are needed for the replication process. So that's why they're dependent on a host. The host provides those enzymes for it. Okay, the host cell, in addition to replicating the DNA, also is going to go through transcription and translation and make more proteins. So it's going to take the viral DNA and make mRNA. That mRNA is what codes for those capsid proteins. And so the cell that is infected is also going to generously give of its amino acids and RNA polymerases and ribosomes and all that machinery that the virus doesn't have, the host cell is providing. So tRNA, rRNA, RNA polymerases, all those kinds of things are what the host cell provides in order for the capsid proteins to be made and the viral DNA to be made. So we've got the capsids and we've got the genetic material. We have everything we need to make a new virus. So those parts are going to assemble into new viruses, and they are going to exit the cell. Sometimes they exit the cell peacefully and just go on their merry little way to infect other cells. Sometimes they exit the cell violently, exploding the cell and killing it. And so the book talks about the lysogenic cycle versus the lytic cycle of reproduction. So the lytic cycle kills the cells, the lysogenic cycle doesn't kill the cells. It allows the cells to keep living. And when we talk about host cells, just making sure you understand that some viruses have a broad range of host cells, and they can infect things all the way from insects all the way through humans. Some have a very, very specific range. So some can only infect certain species, and some can only infect certain cells even of certain species. So HIV only infects the CD4 T cells of a human or a higher primate. But it only affects those specific cells. The cold virus only affects cells of the upper respiratory system. It's not going to affect your liver. Okay, rabies can affect any mammal. So if there's not, some are broad range and some are specific in terms of host cells. How many minutes do I have? 4. We're good. Okay, this is showing you what it looks like if it's an enveloped virus. So remember that envelope is pieces of lipid membrane that came from a host cell, and it's got those proteins on the outside. This is showing you an RNA virus that's enveloped. Again, we're very familiar with the enveloped RNA viruses these days. So these glycoproteins, so they're proteins that have specific carbohydrate markers on them. They bind specifically to receptors on a cell. So they're going to bind to receptors on the host cell, and the host cell is going to say, hey, come on in. It's like they're knocking on the door and you let them in. Okay, you'll notice this time what's coming in to the cell. The RNA and the capsid, so the full virus is coming in. The envelope's not coming in, but the whole virus is coming in, the capsid and the genetic material. Again, the genetic material is the capsid's going to be broken down so that you have this viral genome. Here it's an RNA. And there's different ways that the RNA can be used. This is showing you the viral genome of the RNA being used as a template. So the viral DNA is this red one, and that's the template, and then the cell is going to make complementary RNA. Okay, so it's using this as a template, so if there's a C in the template, what's the cell going to put in? A G, because it's complementary. We're talking about RNA, so we have, what letter replaces T? U. U bonds with A instead of T bonding with A. So this viral DNA is RNA, sorry, is going to be the template, and again, we're going to make two different things. So the template that makes this complementary RNA, the complementary RNA is going to function as the mRNA. So you're not going directly, like you're not using the viral RNA directly to make proteins. The cell makes the complementary one first, and that complementary one is what's used to make the proteins. Okay, so this is complementary. The complementary mRNA acts as the template for transcription and translation. So that then can be used two different ways. It might go to the ER or the Golgi and be used to make these glycoproteins, which are going to go on the envelope. Or in the cytosol, the RNA and the ribosomes and the tRNA are going to make proteins out of that. So the mRNA is used two different ways. Then it's also used, the complementary one is used as a template to make more RNA. So it's like a two-step process. We have the viral RNA, the complementary one is made, and then the complementary one of the complementary one is like the original. Does that make sense? So if we had a sequence of, we're going to go really simple, CGC was the original, then the cell is going to make a complementary one, which would be GCG. So that will be used to make proteins, etc. But then the cell is also going to copy that, make a complementary to the complementary one, and that's going to be the actual, like the original, that's going to be the RNA that's needed to make a whole new virus. So we've got proteins, we've got genetic material, and we've got the glycoproteins for the envelope, and this whole process, assembly process, then is going to kind of bulge out or bud out, which might remind you a little bit of exocytosis. The virus is just going to escape out, again, not killing the cell. That's where we're going to stop. We will finish talking about viruses on Wednesday, and we'll start right in on chapter 27 with bacteria.